My name is Nalini Prasad. I am working for Sugar Research Institute of Fiji in Plant Pathology Lab as Senior Scientific Officer. My role and responsibility is to look after sugarcane diseases, and today I'll be telling you about one of the important disease which was identified in 2004 and confirmed in 2006 that the disease is present in Fiji. That is ratoon stunt disease. We also call in short form RSD disease. So in this disease is caused by a bacteria called Liphozonia zali. What happens, the bacteria multiplies inside the sugarcane stalk, and it blocks the xylem vessels of the sugarcane stalk. That xylem vessel is playing a very important role inside the stalk, where it takes the water, food and nutrients from the roots and up to the leaf for photosynthesis. When the bacteria is concentrated in the xylem vessel, it's not the activity, the process of food making, the leaf is unable to perform, and that causes the stunt growth of the internode. So what we do, we go to the field and we identify the stool that is infected by picking the stalk that is active in a stool that actively growing by showing other stalks are stunned. So what we do, we cut these stalks from the base and we bring it to the lab, and we cut these stalks into small pieces at an angle of 45 degrees to extract the sap to analyze for RSD bacteria. And the sap is extracted using the vacuum pump, where we take the sap when we collect the saps, we label it. We label the sap according to the farm number, and then we bring it to the lab. This is how we bring the samples. We store the samples in the lab, and then we do the lab analysis. We use microplate for analysis, and this is what we call microplate. With the help of multi-channel pipetta, we put the sap inside this plate, and the empty wells we cover it with the coating buffer. And then we take the plate to the imino washer. Imino washer will wash the plate and give us the positive and negative samples, from which we take the samples to the microplate reader. In the microplate reader, we analyze the samples, and the machine will generate and give us the positive and negative samples like this. So that is how we identify a field is infected with RSD bacteria or not, a field is positive to the disease or not. The other disease that the plant protection is focused on currently is Fiji the leaf scald disease of sugarcane. Leaf scald disease is caused by the bacteria, and recently we have found that most of the field Nendiri variety is infested with this disease, This disease is associated with climate. As the climate changed, we have found this disease prone in the field. So in past, we were considering the leaf spot disease as a minor disease, but since 2018, we are coming with the incident that was very small, and now all of a sudden the incident came up to 20% for sugar industry. The field are infested, 20% of the field are infested with this disease. The symptoms of the disease is the first phase of the symptoms, the leaves will show pencil-like structure. The second phase of the disease is the bleaching of the leaves. As you can see here, the leaves have all bleached. The green portion is all disappeared. And as you all know, the leaf plays a very important role for a plant in making the food. Since the bacteria is concentrated inside the sugarcane stalk in xylem vessels, the roots are unable to absorb the water and the nutrients from the soil up to the leaves to photosynthesize, and this end up causing the plant instant growth, and by this the plant will it will dry up, where the plant won't be able to cope and the growers are having the loss. You can see this stool is heavily infested with the leaf scald bacteria, and the other symptoms you can see there's the heavy infestation that the plant, the stalk is able to tolerate, but causing the side shoots to come out, and this will reduce the yield and also the weight return of the sugarcane. To manage this disease, how to control this disease, the water treatment that the institute is having. So we recommend the growers to take the treated seed materials from the sheep plot to cure this disease. That's the only option we have to treat the bacteria on sugarcane.
Thank you so much to the Sugar Research Institute of Fiji for having us once again. Coming up next, we visit a nursery in Cornivia to see how they build up their business and success in this field.